when adjusted by dose amount, with over 2 billion doses of the AstraZeneca viral vector-based vaccine already produced. These are figures from December 21. So, looking to that COVID-19, let us see how or what we have done or why today even the earlier speakers also told that India has become the pharmacy of world. Why? Supplies over 60% of global demands of vaccine, 40% of generic demands in US, and 25% of all medicine demand in UK. You can see a huge potential of pharmaceutical exports. India has more than 10,500 pharmaceutical manufacturing units that caters to demand from more than 150 countries. That means we are talking about almost 75 to 80% of the world, with many that are compliant with US FDA, WHO, GMP, Medicines and Healthcare Products Regulatory Agency. So the major ones are covered. Indian pharmaceutical industry is third largest by volume and 13th largest by value. But this 13th largest by value in maybe next 2 to 5 years we will again be in top 5. Volume wise we are, but value wise also will reach there. Largest generic drug provider. Pharmaceutical export from India was US dollar 20.7 billion financial year 2021 as compared to 19.14 billion in 1920. Although we say our economy was going down, but still pharmaceutical exports have increased. Low cost of production, why is this happening? Availability of skilled manpower, low cost of production, and in-house R&D increased efficiency of Indian companies. And this is bound to go upward only. Our pharmaceutical industry will develop much more at a much faster pace in much more variety of products, and that is where we will definitely in next 5 years be in the top 5 countries by value or volume. But then this requires some changes in the pharmaceutical industry. So I went through the reports that have been given by Deloitte, Ernst and Young, McKinsey, and then I came down that after reading, analyzing, discussing it with my faculty colleagues at our pharmacy college, then there are three main things that needs to change: manufacturing strategy, supply chain disruptions, and increased use of digital technologies. We'll look one by one into each one of this. 4 best practices for pharma companies to follow in developing a successful, sustainable manufacturing strategy. Create business-driven aspirational goals. Put business first. Network strategy design should relentlessly focus on business requirements. You alone cannot do everything. So that is where the networks. Manufacturing strategy that has established the business need and guiding principles can go back to them in times of misalignment, leadership pushback, or critical decisions. This can include setting guardrails around the time frame for payback, such as a break-even within 5 years or limitations on when a brick and mortar expansion may be considered. So more and more network would help to create more business-driven goals. Be radical but not incremental. It can be risky to move historically important or flagship sites, but companies that allow every option to be considered, think holistically and make step change decisions versus incremental decisions are better positioned to achieve transformative change. So the scale at which we work, the speed at which we work, that needs to change. Changing and implementing a manufacturing strategy is a significant undertaking, even if it only involves small adjustments. So why not for economies of scale by doing it? Radical changes such as major site moves can bring outsized results. Second, make strategy design comprehensive. Make operation strategy an input to network strategy. Holistic approach to manufacturing strategy factors in all the elements of operations such as make or buy. Either you make at your own plant or maybe you buy it from outside. The variety of the products that you manufacture. Technology outlook, value chain of the future, not just manufacturing sites. Right from the R&D lab till the place of use. That is what we call as the value chain. So that whole has to be comprehensively looked into. Therefore, the manufacturing strategy needs to include all the internal, for example, internal sites and external, for example, contract development and manufacturing organization. So you see this in a totality. Aspects of the manufacturing network and the manufacturing strategy design and detailed planning process needs to accommodate strategic partners. Segment the manufacturing network as per your product profiles. As supply chain becomes more complex, the manufacturing strategy needs to be tailored to each value chain. For example, one company with a diverse portfolio differentiated in manufacturing strategy so that large capital equipment would be manufactured closer to customers, while manufacturing locations for small goods were more flexible since they could be shipped long distances more easily. You will save on time, you will save on cost, and you will achieve higher efficiencies. Think beyond total landed cost. Best manufacturing design used to be dictated by total landed cost, but today to deliver full value, organization must consider many more factors such as risk, resilience, supply chain responsiveness to determine risk-adjusted total delivered. For example, regionalization has become a key concern since the pandemic has exposed the limitations of global supply chains because they were disrupted. Lot many things were not coming as per the schedule. So that is why you need to go for this. Also, sustainability increasingly at the forefront of strategic supply chain decision making will have both immediate and long-term implications. Use analytics and solutions scenarios to support selection. Take time to get the facts. It is critical to collect good data and align on the fact with relevant business leaders to ensure that the initiatives are credible. For example, Burn organization pushed to move quickly into analysis, but then lost weeks aligning the organization on the conclusion to its analysis because leaders question the validity of the underlying data. Sometimes you do not need to waste more time into data analysis. Instead, have some minor analysis and just take the decision quickly. This not only jeopardizes timelines but also damage the credibility of the decision process. Let's start broad and quickly narrow down. It is best to start with a wide funnel of different solution scenarios, then rapidly narrow down to those predicted to have the highest potential. Nowadays, lot many models are available by which this can be done. Scenarios can be created, and you can come down to the optimum model that you would like. This allows many possibilities to be considered and increases the chances of converging on the best. Then we look into the manufacturing strategy part. Support business-led trade-off decision making. Manufacturing strategy decisions can often be emotional but bureaucratic. Framing key trade-offs with fact and analysis can help combat bias, as can assigning people to challenge preferred options before they are able. So, four systematically manage. You can have four different choices by which we can have this change. Num- Number one is systematically manage execution. Create a dynamic roadmap, not an answer. Create, don't give a total solution. Have a roadmap, starting blocks. Manufacturing strategy is a long-term global scope and exists to manage a dynamic, ever-evolving situation. Dynamic roadmap that leads to long-range planning and is flexible allows an organization to adapt changes during the roadmap's 5 to 10-year lifespan. Okay. It has to be, but the roadmap needs to be dynamic. A company should also define triggers for when the strategy should be reassessed and refreshed, such as in the following instances. When major changes occur, such as in tax, regulatory, and product or program, that may require significant moves or a new site as well as a refresh strategy. Then create a dynamic for going ahead. Institute a rigorous stage gate process. Stage gates are common in process like R&D. Many organizations do not use them to manage manufacturing opportunities. R&D can take a time, but utilize that time to do better things or start the planning for the other functions. A clearly defined and communicated stage gate process ensures alignment at the level of detail required to progress to subsequent stages and limits the tendency to redo analysis and revert to prior stage after a gate has been passed. So eliminate this. The stage gate process can be used to benchmark and measure annual tangible goals, provide a sense of progress, and combat a lack of urgency that can sometimes affect the long-term effort. Integrate moves into the profitability of sending and receiving sites. Measuring the profit and loss implications of transitions ensures that the sending and receiving site leaders focus on the transfer as if it were a part of their business. This will help ensure timelines are met and resources coordinated to support the handover activities necessary for success. While this requires facility leaders to implement significant change management practices, it also gives them appropriate accountability and control. The thing is, as you go in for the integration with profitability, also identify where are your manufacturing, where are your receiving, all those located. Based on that, just integrate the whole process. In the past 2 decades, the worldwide value of pharmaceutical goods traded has grown sixfold from $113 billion in 2000 to 629 billion in 2019. Amid this growth, supply chains have become increasingly global, complex, and opaque. More companies are outsourcing production to contract manufacturers, adding new modalities such as cell therapy, exploring novel ways to reach patients. For some products, this results in supply chains that are so complex that they start in Asia, maybe India or China, and circumnavigate the globe twice. This also happens. Disruptions like natural disasters, international trade tensions, cyber attacks, and global pandemics are just a few of the shocks that can immobilize pharma. So we need to have supply chain disruptions in the post-COVID era that can make our whole supply chain efficient. Four ways to build this supply chain resilience. Supply chain resilience requires four elements: end-to-end transparency, routine stress testing and reassessment, reduced exposure to shocks, and supply chain resilience on the executive agenda. Start with the first one, end-to-end transparency. Lack of visibility into the business practice of suppliers can be a significant risk for pharma. You cannot overlook anybody in the value chain. Company must map its suppliers by tier to have an end-to-end view of the supply chain and identify vulnerabilities. It's also vital to have a clear understanding of exposure beyond supply, including how products are developed, delivered, and stored, because each stage poses its own potential problem. Especially when contract manufacturing is there, this thing is of utmost importance. Routine stress testing and reassessment. Companies often use scenario planning and simulation models to anticipate their vulnerabilities, quantify the potential impact, and mitigate the effects. For example, during COVID-19 pandemic, leading pharma companies use a digital team simulation to understand the impact of production slowdowns and shutdowns on the supplies of patients' medication. This helped the company realize that it had more time than it had anticipated to design and implement safer ways of working at its manufacturing plant, allowing it to take more time to get the best solution. Once a company has visibility into its supply chain, it can assess the likelihood of different risk continuously. Third, reduced exposure to shocks. Relying on a single source for critical component or raw material can be a vulnerability, as can depending on multiple suppliers concentrated in the same place. A company can also strike a better balance between just-in-time and just-in-case inventory levels, harden its physical assets to withstand hurricanes and storm surges, and provide financial support to distressed but essential suppliers. Lot many times your supply chain can be broken if you rely on only one, but relying on more is also a problematic because that increases your cost, increases your time, decreases your efficiency. So you need to have a balance between both of these. Supply chain resilience on the executive's agenda. Risk and resilience should be embedded in an organization's strategic planning and day-to-day execution with structured governance to ensure that decisions are made and acted on at the right level in time. The planning department identifies the decisions to be made. Lot many other departments implement this, but after implementation, what the results are, that again needs to be given a feedback to the planning department so that they can change on a dynamic way, on a day-to-day basis, or maybe week to week, month to month, or a quarter to quarter basis, rather than waiting for a longer time. And then ultimately, the use of digital technology. The world has seen in this pandemic how digital technologies were used in each and every field, be it entertainment, education, manufacturing, pharmaceutical, whatnot. So that is where significant investment in technology and artificial intelligence will lead to true digitalization of value chain from R&D through to commercialization and including the use of omni-channel marketing. Utilize each and every channel offline or online that are available for marketing, positively impacting drug discovery, digital clinical trials, and interactions with healthcare professionals. Now, the thing is, digital technologies are lot many, and a company will need to identify what can be the best use of whether it is artificial intelligence, data science, machine learning, or any other mode of this digital technology. You need to customize your solution, and that is where it is an altogether different technological domain, which is beyond my purview to explain here. And then ultimately, this pandemic has given the pharma leaders of the pharmaceutical industry to ponder upon not many points. These are few of the points that I have identified. What is our organization's contribution toward the overall effort to combat the virus? Be it vaccines, treatment, testing, whatever you are into. You need to know how your role can be. What was your role? Can you still improve it if the next pandemic comes? How can you utilize the opportunity to become a more maybe a leading company or a global company? Can we use advanced analytics and modeling techniques to help model scenarios in R&D and supply chain operations? The time to do R&D and ultimately come out with a drug in a commercial way. Can we decrease that? We have already seen with vaccines. Can we replicate this with others also? What is the impact on our sales force in a virtual environment when you are not meeting your sales staff? They are not meeting their clients or customers, but still company's targets needs to be achieved. So how is all this possible? So can we improve on the virtual environment? How will our existing and planned clinical trials be affected? Can we do the clinical testing in the same manner or we need to change? What is the best approach to conducting and maintaining a supply chain risk mitigation and interventions assessment? Whatever we were doing pre-COVID in supply chain risk assessment, should we continue with the same thing? Or now the risks are more and time is less? What changes do we need to make to our business model to adapt to future scenarios and crisis management? Pandemic was a crisis for every healthcare company and crisis which neither the company nor its counterpart nor its competition had ever seen. But in spite of that, everybody identified a model, a solution to that. So now that we know that some type of pandemics may come, are we more prepared for the next pandemic or not? That is one of the important questions that we need to ponder. And then what is international best practice in supporting our people working in a virtual environment? Like e-learning or teaching through online mode or having meetings online. Be it any of the platform, Google or Zoom or Microsoft Team or whatever. That was unknown to us. But the pandemic taught us very fast. It was a knee-jerk reaction. All of us learned, right from like what Sir told, a 5-year-old kid to 80-year-old person also. Everybody learned how you can do this on Zoom or Google Meet or whatever platform. So that is where how do we do this? How do we create advantages with the virtual environment? And what changes should we implement to mitigate risk with the disruption of non-COVID-19 related clinical trials? COVID-19 is one, but what about other things which are not related to that? How do we take those things? And then how do we work with our key customers, suppliers to fulfill demand in current high peaks, as well as partner to sustain their reach? When the second wave came, the shortage of oxygen all across the country, how was that met? So can we do it much more efficient still? That is what the pandemic has taught us. So maybe oxygen was one thing. There can be lot many other things. Yeah, but yeah, remdesivir and a lot many other drugs. As a wonder drug, whatever it is, can we do it? So these were few of the things that I had identified for this particular session. Yeah. Thank you. Any questions?
What do you think of? We will have the same COVID pandemic again or some other pandemic? Which is not COVID.
No, I think my view says that we will be better planned now. Because the thing is, the world as a whole, irrespective, right from the political leaders of the country to the whole healthcare industry, as well as the general population or any other people who come in the supply chain network, everybody has combinedly fought this war. But okay, major part was done at the strategic level by the political leaders and at the implementation level by the healthcare industry, but everybody has fought. And so that is where we all have learned a very good lesson. So a new pandemic will not be this, means the catastrophic effect will not be at this level. And now more and more technologies are at our disposal.
People will also adopt it.
Yeah, people will definitely, because everybody has done. It's a like the generation which is hardly 5 years, 6 years old today will be living, let us say, for another 75 years. They also know of this COVID. So if you talk about that, at least in next 50 years, I don't see the effect of next pandemic would be coming at this level. This would be my answer.
Which country fought it best?
Difficult question, but not just because I am in India and I am a patriotic, but yes, just look at the complexities with which we work. And a democratic country, being a democratic country, unlike China, China again you can see right now they have got lockdown. But we as a nation, just 2 days back I read Maharashtra and there was this another they have masks are no more mandatory. Yes. So I think with this huge population, number of vaccines delivered to Indian population is much more than number of vaccines delivered in any of the other developed countries, including US. They itself, as compared to the population, the percentage wise is much lesser. In spite of having lesser number of hospitals or any other, what you can say, number of doctors or any other paramedical staff.
Poor medical care.
Yeah, yeah. In spite of that, and the thing is, time was less, resources were also less, monetary wise also. But in spite of that, like we were not knowing what a PPE kit was, and during that pandemic, we became one of the major exporter of PPE kit. Exporter. So that means we had already which met the demand, domestic demand, in spite of that. So these are my arguments by which I would say India has. China as a country may have, but the political system itself is different. So that is why taking decisions is very easy over here. But in fact, hats off to our honorable Prime Minister. The bold decisions that he and his team took, whosoever were there for his advisors from healthcare or from any other sector. I think I can proudly say yes. Today, means we fought the battle better. Although I lost my father in this pandemic, but still I would say as a country, India has fought it much. Yeah. Yes, sir.
What I feel is this pandemic has converted into an endemic. अब किसी को आ गया.
Yeah.
Am I right?
Yeah.
Omicron आ गया.
Yeah.
So it is like a natural vaccination.
Yes.
India में the way the rallies were there.
Yeah.
What is your opinion? Has it not spread there? It must have spread. So endemic case हो.
Yeah.
Natural vaccination हो गया.
Yes.
Now China is struggling.
Yeah.
To control. अरे क्या control करना? हमारे यहाँ तो सबको
Yeah.
already 175 crore हो चुका. Plus endemic. So I think Corona का नामो निशान मिट गया.
Yeah.
Am I right?
Definitely, sir.
Am I right?
Even whatever reason government was definitely, sir.
But correct है मैं जो बोल
Yeah, I told you.
Yeah.
We are free from fourth wave of pandemic.
We are free.
We are free. We want a fourth wave.
Whatever precautions we had to take as a population, I know how disciplined we were. All of us knew. But in spite of that, the effect was lesser.
Sir, Omicron सबको आके गया.
Yes.
Am I right?
Yes.
सर्दी खांसी किसी ने टेस्ट नहीं किया.
हाँ.
3-4 दिन रहा.
Yes.
Finish हो गया.
Yes. We are out of this.
We are out of it.
Definitely. And now that booster dose is also going on, so I don't think we can have anything like this, at least in near future. Yeah, now we have to work more
At
on
least
it.
हम ये कर पाए.
Yeah.
ये वाला. Operant.
Yes. That is the essence. Yeah. Thank you.
Thank you so much, sir, for the session. Now, I would like to invite Mr. Vikram Chaudhary and Dr. Chanik Chaudhary on the stage to present Momentum to Check. Now, I would request sir and ma'am to present Momentum to Mr. Ashok Bajaj. Thank you so much, sir. Ma'am. Now it's time for e-poster session. And to start e-poster presentation, I will hand over the session to Dr. Juhi Pradhan. Hello. Very good afternoon, everyone. So we are going to start with the e-poster presentation, and we are having approximately 10 to 12 presentations here. The first presenter who is present with us is Parul Aiman. Good afternoon, one of all present here. I am Parul Vaishnav, student of Indore Institute of Pharmacy, and my topic of presentation is formulation and development of 3D printing based tablet dosage form of phenolphthalein. Well, 3D printing is a rapid prototyping method which creates 3D objects by depositing several layers in sequence. And we can design this 3D object with the help of AutoCAD software. 3D printing technology is also known as additive manufacturing because there is an addition of material, and it is also known as layering technology since the addition is in the form of layers. Nowadays, pharmaceutical 3D printing is gaining a considerable attention as a potential technology, enhancing efficacy, preciseness, and individualization while reducing wastage cost. Well, we all know that Conventional dosage form has been used for a very long time for treating patients, but hygroscopic drugs, drugs with amorphous in nature, drugs with poor water solubility, slow dissolution, and the drugs which have high absorbance in GIT are difficult to formulate in tablet. But 3D printing is one of the most powerful technology which provide unique offer to the drug products when compared to traditional method. It has capacity of producing personalized medicine with flexibility. It can also alter size, shape, and drug release characteristics just by designing it into AutoCAD software. While keeping these benefits in views, our aim was to explore 3D printing technology to formulate solid oral dosage form, which may enhance patient compliance and reduce manufacturing costs. There are various types of 3D printing technologies like fused deposition model, stereolithography, selective laser sintering, and inkjet printing. Fused deposition model is one of the 3D printing technology which is available in open source that makes this technology more affordable than others. And I have used this fused deposition model for my research work. For the fabrication of Synarizine tablet, I have used Synarizine 50 mg as an API and polyvinyl alcohol 470 mg as a polymer. Now the 3D printing of tablet was prepared by these steps. First is designing of tablet structure with the help of AutoCAD. AutoCAD is a software. For all the 3D printing technologies, first we have to design our tablet structure into AutoCAD software. We should we should have an idea that what kind of structure we like to print. We should know the dimension, thickness, diameter of the tablet. Then we just have to design it into AutoCAD software. Then once the design is completed, we go for the slicing of the tablet with the help of Cura Ultimaker software and then converted it into STL file format. We can install this Cura Ultimaker software and AutoCAD software in our laptop and computer also, and it is very easy to handle. Then the STL file format that we have generated is converted into G code. G code is actually numerical coding programming language. As we all know that computer can only read a specific language. That's why STL file is converted into G code. Then we prepare our FDM printer. First, we add the filament to the printer. Here I have added polyvinyl alcohol filament to the printer. Then the STL file that we have generated before is copied to the SD card, and that SD card is inserted into the 3D printer. Then the printer starts printing following the commands of G code. G code actually commands the machine where to move, what part to follow, and how fast to move. That's how printer starts printing, and it prints until the solid objects are created. Here I have printed first the holo tablet. Then then I paused the printer. The Synarizine drug was added in a powder form. Then I resumed the printer, and the printer covers the whole tablet. And then I remove the tablet from printer. By this, we can manufacture also multiple design tablets at the same time. And for to check the compatibility between drug and polymer, because I have used here polyvinyl alcohol, so we have to check the compatibility also that drug is and polymer are compatible or not. So the DSA data showed that there were no detectable interaction between drug and polymer. And the Synarizine tablet were also evaluated for weight variation, friability, hardness, and drug content. And all the results were within acceptable range as defined by Indian Pharmacopoeia. Percent drug release was found to be 96.8% at 200 minutes, and this works validate that the FDM, the fused deposition model 3D printing, was capable of producing sustained release tablet. So here that's how I print my tablet in very simple way. And here I would like to conclude my research work that fused deposition model has been widely accepted in the manufacturing of component. It is actually utilized in manufacturing of biomedical device during COVID-19 pandemic also. And the research has been increased after first 3D printed drug approved by the FDA. And here I printed this drug and evaluated with several parameters, and all the results were within acceptable range as defined in pharmacopoeia. So I believe that this was a significant step forward 3D printing technology in medicine, particularly in personalized medicine. Thank you so much, and thank you for giving me this opportunity.
Well done, Parul. You mentioned that you yourself have printed the thing. Okay?
Yes, ma'am. With
In
the
your research laboratory?
design, actually, I went somewhere in Indore,
Okay.
and they told me about how to design.
All the facilities required for 3D printing were there.
Yes, ma'am. Yes, ma'am.
Okay. So I just wanted to ask you some questions. That you asked that 3D printing can improve patient compliance.
Yes,
So
ma'am.
as compared with the traditional method of tableting, how can you say that this can improve patient compliance much more than conventional?
Ma'am, take an example of Cinnarizine. Cinnarizine is widely used for motion sickness, and the dose of Cinnarizine is two tablets 3 times a day. But here I've given 50 mg tablet and I've prepared sustained release. So you have to take one tablet only, one day, per day. So it improves patient compliance. It
But
reduces
yeah,
multiple
you
doses.
you are right, but it can also be done with conventional method of tableting.
Yes, ma'am.
We can also prepare a sustained release tablet by that method also.
Ma'am, but it reduce time and cost also. Manufacturing costs are also reduced.
Anybody who want to? Yeah, yes.
तो आपको ऑपरेटर का इंटरनेशनल आने वाले के साथ आप आने वाले के साथ चला तो आपको कैसा
Very good, sir.
So well tried. Try to study more about the release pattern and the advantages of this method of preparation of as over the conventional. Okay.
Thank you.
All the best. Next presenter. Next presenter with the code EP102. Is he or she present here? EP105. Prachi Gupta. Is she here? Come and present.
Good evening to all present here, respected sir and ma'am. I am Prachi Gupta from Indore Institute of Pharmacy, Indore. Today I am here to present an overview of 3D printing technology in the pharmaceutical field. So first of all, I am coming to the introduction. So 3D printing technology is a process of fabricate a 3D objects where successive addition of materials in the form of layers using computer aided design software. The material is deposited in the form of layers. That's why this process is also known as a layering technique. And here we use the addition of materials to form objects. That's why this process is also known as the additive manufacturing process. Now, the benefits of this technology, so when we compare to the conventional method, it has a lot of benefits. First, flexible design. Second, we can print on dosage form is based on patient requirements. And next is it gives a fast design and protection. Now, the major limitation of this technology is a reduction in manufacturing jobs, and another limitation is a copyright issue. Now I have taken a few techniques of 3D printing technology. First is selective laser sintering, stereolithography, inkjet printing, Windows technology, and the last is the fused deposition molding. Now I have various applications of 3D printing technology in the pharmaceutical field. First is the bioprinting of tissue and organs. Next, we formulate a unique dosage form. Third, we customize the drug dosing, and the next is complex drug release profile, and we customize the implants and prosthetics, and the last is the anatomical model for surgical preparations. Even during the current COVID-19, this technology is very helpful for preparing the face mask, ventilators, respiratory walls, and also nasopharyngeal swabs for diagnose the COVID-19. Now the materials and methods. In the pharmaceutical field, the 3D printing requires special plastic that is easily melt and adhere. These plastics are also known as a thermoplastic filaments. Like an example that I have mentioned here, first polyvinyl alcohol, polylactic acid, polyethylene oxide, and high impact polystyrene, etc. Now I am coming in the method that how to design a tablet using the CAD software. Fast design and production. These are the some driving factors which is responsible for growth in the market. Now I am here to conclude this review this technology is a very valuable and potential tool for the pharmaceutical sector because it offers the personalized medical medicines focused on the patient needs and it gives numerous benefits such as increasing the cost efficiency and manufacturing speed. And now this technology has given a wide range of applications. Like some examples, we have print tissue and organ with the help of this technology and it gives a customized dosage form. Thank you so much.
Okay, Prachi. Well, right. So yours is the review one. Okay.
Yes, ma'am.
You told us about the 3D printing. I just wanted to ask you, can you name any organ? You just quoted that we have printed tissues and organs. Can you name any of the organs which has been printed from this 3D printing?
Yes, ma'am. Ma'am, heart organs and liver organs, these organs are also printed with the help of this technology.
Other than pharmacy and biomedicines, any other use of 3D printing?
Yes, ma'am. Ma'am, in other sectors like aerospace and automotives, ma'am, is also used in a textile industry and it also used in a.
Oh, okay, well, thank you.
Thank you.
Our next presenter is Mayank Pradhan.
Very good afternoon to all of you. My name is Mayank Pradhan. I am from Computer ISET College of Professional Studies and Pharmacy College, Indore. My review paper topic is drug used to treat COVID-19. Okay. So now the first case of COVID-19 was reported in year 2019 in Wuhan seafood market, China. On 11th March 2020, the World Health Organization declared it as a global pandemic. Till today, more than 46 million cases are reported, and from which more than 63 million people are died. Now, what are the general symptoms of this disease? The general symptoms are fever, fatigueness, shortness of breath, hypoxia, and etc. Through this review, I just want to give the presentation about the all the substitute drugs which were used to treat COVID-19. Mainly, all the drugs work on the basis of three mechanisms. The first one is receptor binding. The drug get attached to the ACE2 receptor and TMPRSS receptor 2 on the host cell. The second mechanism is it acts on the viral RNA and viral proteins. The third mechanism is the drug acts on the viral structural proteins to destroy. Mainly from all the pharmaceutical agents, mainly the antiviral drugs and immunomodulatory agents show their potential action against this disease. The drugs are azithromycin, which act as a mimic, binds to the receptor and inhibit the binding. Second is doxycycline, act on the MMP9 protein. It is a structural protein of virus, so that the virus can kill. Ivermectin, it acts on the viral RNA. Hydroxychloroquine, an anti-malarial drug which inhibit the viral enzyme and processes. Dexamethasone, it acts as a mimic to produce inflammation naturally into the body so that our defensive system can awake. Favipiravir, the second most famous drug for this treatment, it halts the viral replication. Clindamycin, it get attached to the receptor of the host cell and then inhibit the binding. Remdesivir, the most famous drug used during this treatment, it inhibits the RNA-dependent RNA polymerase. Now, our DRDO invented a drug. The name is 2-deoxy-D-glucose. It enters the glycolysis cycle into the virus so that the virus can't grow. Other antiviral drugs such as molnupiravir, lopinavir, ritonavir, and ribavirin. These drugs inhibit viral replication and the viral RNA enzyme. The doses of drugs are mentioned here, and from all these drugs by the WHO, FDA, and US FDA, azithromycin, ivermectin, the use of hydroxychloroquine, favipiravir, remdesivir, lopinavir, and ritonavir are banned for the treatment of these diseases. Because these drugs have their own side effects, these are not the proper treatment of this disease, they are just as a substitute. Now, the life cycle of SARS is classified into the two stages, the initial one and the advanced. Several viral and host proteins are important for each stage. The treatment through these antiviral drugs and immunomodulatory agents are available, but these are only substitute drugs for the mutant. Now, I want to conclude that from this review, the potential therapeutics that interfere with these some events of viral cycle. Important drug targets at this stage are as spike S protein, ACE2 receptor, TMPR SS2 receptor, viral replication, viral enzymes, and proteins. The vaccines are necessary for everyone and they are effective against this virus. But a proper and fixed therapeutic dose for COVID-19 are still a part of research and pharmacology. Now, the virus is doing mutations by changing the sequence in its viral RNA. So, it is difficult for the scientists also to discover a 100% effective therapeutic agent against this disease. As I say, the antiviral drugs and immunomodulatory agents such as vitamin C, zinc, they also play a very vital role for improving our immunity. Methyl or corticosteroids, they are also used during this treatment. Plasma therapy is also used, from which plasma therapy and zinc are also done for the treatment of this disease. After the injecting remdesivir, clindamycin, many of patients experience cardiological issues such as coagulation of blood. For that, they were injected heparin for the vaccines still have unknown results at some extent because the clinical trial was so much alterated and they are still a part of it. Thank you.
Okay, Mayank, very well presented. Right, well. So you have mentioned a lot many drugs used for the treatment of COVID-19.
Yes.
Can you say that they were able to treat the pandemic or treat the disease, not pandemic? They were able to treat the disease or just try?
Ma'am, actually, a proper therapeutic agent is still unknown. So, the medical staff, the doctors are just doing a trial with the antiviral drugs and immunomodulatory agent.
By understanding the etiology of the
That is
virus.
so that what the result they get, according to that, they prescribe the drug.
So how can we design some specific drug for the treatment?
Sir, ma'am, if virus stop the mutation, it will surely.
You need to design the specific drug.
Ma'am, actually,
Although you are having a lot of targets, the targets you have mentioned in your presentation.
if we design a specific drug,
So what should be the target for these antiviral drugs, anti-SARS-CoV-2?
the main target should be receptor binding and the damage of viral RNA.
Yeah, so these are the proteins specific to SARS-CoV-2. So we can target these proteins and design some new drugs with the help of CAD. That means CAD, CADD. One of the presenters told previously. What
AutoCAD is a software.
computer aided drug. Okay? All the best.
Thank you.
So the next presentation is of Satyam. Satyam Sangha.
afternoon to all the dignitaries present over here. Myself, Satyam Kanwan, in front of you to present my review paper uh, by a e-poster presentation, which is on the topic diagnosis and treatment of COVID-19. Coming to the introduction, COVID-19 rise from Wuhan, China in 2019, 96.2 genomic sequence similar to that of BET and 79.5 similar to that of SARS-CoV. So it's officially first designated as 2019 nCoV and it's laterally revised as uh, acute respiratory syndrome coronavirus 2 by its genomic sequence change. Coronavirus family can be classified into four types on the basis of their mutations, which are alpha, beta, delta, gamma, respectively. Coming to the method and methodology, for the diagnosis of COVID-19, there were used two mechanisms, from which first one is viral detection and second one is immunity response. In viral detection method, swab sample is mainly used as for the diagnosis of the coronavirus. There are two biomarkers which are nucleic acid and viral antigen. Nucleic acid used by the test of RT-PCR, which was considered as the best test method for the diagnosis of coronavirus COVID-19. We are also using hybrid methods. We are also using DNA sequencing. Coming to the viral antigen, antigen is being used. We all of are familiar with rapid antigen test, which is done by this antigen test method. Coming to the immunity response, what our body shows response towards the virus comes under this. Coming to the drugs or the treatment used for the COVID-19, from vaccines, from drugs, from convalescent plasma therapy, we are treating COVID patients. For mild patients, we are using azithromycin, doxycycline. For moderate patients, we are using clindamycin, molnupiravir, hydroxychloroquine. Of corona are constantly detecting worldwide. The vaccine play a major role in decrease new number of patients. As seen in the middle 2021, the total number of cases were increasing exponentially. It's increasing readily. Why we can clear that by graph. But coming by after the proper diagnosis and treatment, the graph is been stabilized due to the proper treatment and proper diagnosis. Conclusion through several literature study, it was concluded that various diagnosis method available for the detection of coronavirus. RT-PCR is the most reliable method for the diagnosis of COVID-19 at present. At present, that there are still no effective therapy for the treatment of SARS-CoV-2. Researches are going on. We believe that in a short period of time, effective scientific measures will it will help us to fight with corona. Thank you.
Thank you, Sachin. So everybody knows, I think, everything about coronavirus and COVID disease now. I just want to ask you a very simple question. What is the full form of RT-PCR and how can you say it is the best method for
reverse transcription polymerase chain reaction? Ma'am, it's best method as because we are learning the RNA by converting it into DNA of the virus. So we can clearly we can clearly judge whether the
DNA of the virus?
yes, ma'am.
It is an RNA virus?
No, no, ma'am. Ma'am, we are converting RNA means we are just transferring.
Reverse transcription. Yes, that is known as reverse.
Yes.
Yes. So
when we are just transcription, it means we are just transferring, so we can easily judge whether the virus is present or not. Any other recently introduced method you know about? Okay, thank you very much. Next poster presentation is of Akash. Yes, Adarsh.
Good morning to all. My name is Adarsh Vijay. I am here to say something about vaccination of COVID-19. It is my review paper. In December 2019, coronavirus disease was firstly reported in Wuhan, China, which became a pandemic. It was spreading very fastly, and due to it, there was a very huge loss of lives. And when there was a huge loss of life, scientists decided to find the vaccine against the coronavirus. But they are facing lots and lots of problems. The main reason was that COVID was mutating again and again. COVID is mutating again and again, due to which it is having five variants. After that, on 23rd August 2021, the first vaccine was introduced, whose name was Pfizer BioNTech COVID-19 vaccine. Vaccines generally work on four mechanisms. First mechanism is messenger RNA vaccines. In this vaccines, genetically engineered RNA is used, which goes inside our body and teaches our cells how they can form spike proteins on the surface of the cells. When the spike proteins are formed on the surface of the cells, at that time our body, when detects it, it starts making the antibodies against the cells. Viral vaccines, the second types of vaccines. In this type, we are using a harmless virus, which work as a delivery agent, which in which we are putting the genetic material of coronavirus. When this type of virus goes inside the body, it starts making spike protein by using our cells. When the spike protein is formed and our body detects the cells, at that time it started making the antibodies. The example of this type of vaccine is Sputnik Janssen by Johnson and Johnson. The third type of mechanism is protein unit vaccines. It contains one part of the virus, which is mainly responding on our immune system, which has a highest response of over our immune system. Here in coronavirus, that part is spike protein. We are taking a part of spike protein, which is harmless for our body, and we are injecting it inside our body. When this spike protein goes inside, it does not harm the body, but due to this, our body starts making antibodies. The example of this type of vaccine is epivac vaccines. Four type of vaccines are whole virus vaccines. In these types of vaccines, we are using weak viruses. We are making our virus very much weak that it cannot harm our body, and we are making them weak by using heat or chemicals or radiations. And when we are injecting them in our body, the virus does not harm our body, but our body starts making antibody against it. So that when the original virus comes, our body can fight against it. The example of this type of vaccine is Indian Covaxin. Result: 64% of total world population has been vaccinated till now, in which 60.2% of population of India is fully vaccinated, and the vaccines plays a very important role in reducing the case of COVID-19. Conclusion: in this review paper, I want to conclude that vaccines are effective against coronavirus. Once we get a start of vaccine, our body starts making antibodies against the coronavirus. When the real virus comes, our body then fights with it. But we can say that the vaccine is only for the particular type of virus. For example, now the new version like Omicron is coming. and then they are the person who is fully vaccinated, they are getting infected too, and many people are also dying due to this. So we can say that vaccines are good, but we cannot fully depend on it. Hence, we have to follow the COVID guidelines.
लेकिन अगर सुनकर एक तो आप लोग ये जो प्रेजेंटेशन कराते हैं इसमें जो फोटोग्राफ्स लेते हैं वो जनरली फॉरेनर्स के होते हैं, आप कोशिश करिए कि इंडियन पॉपुलेशन के हो तो ज्यादा अच्छी बात है।
It was a photograph of the first madam who has taken the.
और
ये
चाइल्ड है
नहीं child नहीं है, lady है lady.
लेडी ओके then well done. Okay, so can you mention how many vaccines are available in India right now and which population is currently being vaccinated?
60% population is totally fully vaccinated.
Age wise?
एज वाइज मैडम अभी तो 12 से 18 के बीच की पॉपुलेशन को मतलब वैक्सीनेटेड किया जा रहा है.
And which vaccine is being used?
कोवैक्सीन mainly कोविशील्ड is mainly used.
Covishield or Covax?
Both are used. But
Covax. Anyways, have you heard about Zycovid?
I don't know.
Zycovid is another vaccine which is a DNA vaccine and it has been developed by Zydus. It is also available. Okay.
Thank you.
Next is Mohit Puri.
Good afternoon to all respected shareholders, trustees, and all participants. My name is Mohit Dwivedi from Confident Asset College of Professional Studies, Pharmacy College, Indore. The topic of my presentation is carbon nanotubes in rheumatoid arthritis treatment. Rheumatoid arthritis is a chronic inflammatory. Rheumatoid arthritis is chronic inflammatory autoimmune disorder. In this disorder, body attacks in your own cells due to considering them as a foreign particle. This is this disease is one of the worst population is suffering from them. So the normal treatments is not that much effective. So we focus on the targeting drug delivery system. With the help of carbon nanotube, we focus on that system and it gives us more effective treatment due to potent drug delivery. The methods included these are first, there is no guaranteed treatment. Till now, the research is going on, but no guaranteed treatment for rheumatoid arthritis. From the symptoms and conditions, we are doing research and treating the patient on the basis of their symptoms. First, we use NSAIDs, that is non-steroidal anti-inflammatory agents, which reduce pain. Second, we use corticosteroid. In this, it helps in corticosteroid helps in reducing inflammation. And next is DMARDs, that is disease modifying anti-rheumatoid drug, which helps in slowing process of rheumatoid arthritis and results in saving tissues and joints. And next, biological agents that enhance and modify the treatment, like Aptasat. From this process, we mix them with the coupling like EG, and then they are used in carbon nanotubes to modify the treatment. The methods of RA treatment is when RA diagnosed, then methotrexate plus glucocorticosteroid plus DMARDs. These are the treatment which we can take for 6 months. If there are certain changes, then we can continue the treatment. Otherwise, we have to change the biological agent and DMARDs. Then again, we can change the treatment for 6 months. If the result is desired, then we can continue the treatment. Otherwise, we can change the same the DMARDs and biological agents.
the result is that 1% of the world's population is suffering from rheumatoid arthritis, but not getting the guaranteed treatment. So, with the help of carboxyl and HIPCO single word carbon nanotube, the coupling of MTS, that is methotrexate, it is the immunosuppressant which helps in rheumatoid arthritis treatment. So the level of treatment can be increased. Conclusion is that in this study, HIPCO's SWNB, HIPCO is high pressure carbon monoxide single word carbon nanotube are potent vehicle for drug delivery, which helps in the potential in efficient in the treatment of rheumatoid arthritis. Thank you.
Okay, thank you, Mohit. Can you just elaborate in simple sentence how carbon nanotube can help in delivering the medicaments in rheumatoid arthritis?
Carbon nanotubes are the fourth allotrope of carbon that is Buckminster fluid. So they are a mesh-like structure. So in this we can just put our drug and focus on the target drug delivery. So they just go to the receptor and attack on not attack means they go to the targeted area which we need and they reduce the time of action. And
So are they having specific affinity towards any receptor?
I don't know the particular system, but they are very useful and they are the future of this medicine, carbon nanotube.
You don't know much about
It's a review paper.
it. Anyways, leave it. Can you name any other autoimmune disorder you have heard about? Any other autoimmune disorder?
Actually, arthritis
In which class you are studying?
in pharma final year.
Final year. So you must be knowing.
No, actually, arthritis are of 7 types, and from them,
No,
there are
about
autoimmune disorder. So from them, one type of autoimmune disorder is rheumatoid.
Yeah, that I know. But any other type of autoimmune disorder?
Cancer,
Myasthenia gravis? Yeah. Parkinson's. Myelitis. Yes, yes, yes. Suffering from this type of myelitis. Myelitis means inflammation of myelin sheath. Yeah. Yeah. It leads to paralysis. And sir is such an inspiration for us. A huge round of applause for us. Sir, really, sir, I'm so happy to meet you again. So, okay, thank you, Mohit. So the next presentation is of Praveen Tamar. Not visible to you all. Good morning to one and all. My name is Muskan Haria and I'm from Computer Science at College of Professional Studies, Pharmacy College. And the topic of my review paper is microneedle, a smart approach and increasing potential for transdermal drug delivery system. As you know, transdermal drug delivery system works on skin, like it's related to skin. And when it comes to the drug distribution through the skin, hypodermic needles and topical lotions are the most popular options.
but patients dislike needles because it causes them discomfort and topical creams have low bioavailability. The main obstacle to drug delivery via topical route in the skin is stratum corneum. And as we know, there is a three layer of our skin that is stratum corneum, which acts as a barrier. So, on micro needle, we just pierce it and apply drug directly to the epidermis. Only some molecules such as lipophilic and low molecular weight medication can get to it directly by applying as a topical cream, and the other called the barrier of stratum corneum. So mechanism of drug delivery is like we have an array of we have an array of pads in which hundred of needles are arranged, and it just pierces the skin and from stratum corneum. It just pierces the stratum corneum and the drug is directly delivered to the dermis. There are so many types of microneedle. For example, hollow microneedle, dissolving microneedle, coated microneedles, and the solid microneedles. So they have their own functions of drug delivery. To get the medicine into the epidermal layer, you might employ a variety of approaches. One method is to pierce hole in the skin using microneedles, then remove the microneedle and cover it with a drug containing a patch. This is the work of hollow solid microneedle. Like we just pierce the skin and apply our drug directly on it. So, a direct route for medication to enter the skin is therefore created. For a better effect, the electric field might be used. The second method involves covering microneedle with a drug containing coated layer. This is done by coated microneedle. The drug dissolution occurs when the coated microneedle are introduced into the skin. The third method involves dipping the microneedle in the medication solution and scraping the needle against the skin in the solution. The medication is left behind. Now, result and discussion about this. Silicon was firstly used in the manufacturing of microneedle in 1990s. The research was done to determine whether or not microneedles may be utilized to deliver medication more effectively through the skin. Initially, formulation tests were performed on cadaver skin to see whether a big molecule such as albumin and insulin could pass. So basically, microneedle works on molecules which have a large size. When microneedles were used, further research revealed that microneedles transmit big molecules more effectively. Many new fascinating microneedles ideas are now being developed that will be extremely useful in the future. The microneedle technique is being used on a variety of medication, but it must overcome a number of obstacles before it can be released to the market. Many investigations must be completed before they can be clinically approved. Conclusion of my topic is microneedle are promising new drug delivery mechanism for pharmacological macromolecules, both systematically and locally. The main advantage of this method is its high permeability into the spectrum corneum. As a result, researchers concentrated on the creation of new types of microneedle for alignment such as cancer and heart attack. In conclusion, various patients have been filed to cover the creation of microneedles. This scope is highly beneficial for the next generation since employing microneedles can increase the therapeutic index.
Okay, Vishnu, thank you so much. Can you tell me an example of microneedle-based delivery system available in the market? Is there?
Ma'am, yes, it has a
Direct drug delivery, not possible. drug delivery, any drug which is delivered to microneedle. I don't know. No, it's okay. What is the material of construction of this microneedle? Fabricated from silicon, like we can use silicon for this. And what is the advantages of silicon? Ma'am, it can use in dissolving microneedles and it can get disappear and control the release of them. Okay, a very common question. What is the difference between systemic and topical? Ma'am, systemic to direct veins और मतलब skin के अंदर चले जाती है, topical से ऊपर रहता है. With the skin, systemic जो skin के अंदर चली जाती है. नहीं. Systemic, which enters into the system. Which system? Blood. Yeah, circulatory, blood, plasma to every organ. Okay, that is systemic administration. Okay. Who is ready now? Kriti? Kriti Jain.
My name is Vicky Chen. I'm a computer and a professional study pharmacy college in London. My topic for this paper is vaccine formulation based on mRNA technology. First of all, what is RNA? RNA is a molecule somehow similar to DNA. Unlike DNA, RNA is a single stranded. The primary function of RNA is to create protein through translation. RNA carries genetic information and translate to ribosome and into the protein required for the cellular process. RNA is mainly of three types: mRNA, tRNA, and RNA. Now we are using mRNA. mRNA vaccine. mRNA vaccine is a type of vaccine that uses a copy of a molecule called messenger RNA to produce an immune response. So, very first experiment, mRNA was injected in a muscle of mice and was a successful transcription of a designed mRNA. Ribosomal nanoparticle to cell was published in 1989. The first human clinical trial was for infectious diseases like rabies in 2013, and further studies were done for influenza and chikungunya. And this March 2022, Moderna has announced that there are 15 diseases that can be cured by mRNA vaccines. The most adaptive duration for the mRNA technology was COVID-19. Moderna and BioNTech both approved at the same time. The main goal of the mRNA vaccine is to trigger immune system to produce antibodies that target particular pathogen. That pathogen is targeted is called antigen. mRNA vaccine introduced as short time, synthetically created fragment of a RNA sequence of virus. The fragments are taken by dendritic cell through phagocytosis. This triggers the production of antibodies specifically targeted to antigen. This results immunity. By this, we conclude that mRNA is a promising platform that is very versatile, potent, and streamlined. And the next 5 years for the mRNA vaccine will be most important. Thank you so much.
Thank you. What is your name? Vicky. Your name is not visible from me. Whenever you go for a presentation, beta, you should go for testing yourself first. That the presentation you have made is how it is appearing to the public or to the audience. Okay. The font size you have used, the designing, the graphics, everything, it is not visible to. Is it yes or no? Yeah. So first you should improve your presentation. You have tried very well. At this level, at B.Pharm level, you all are doing very good. अभी ये शुरुआत है आप एम करेंगे आगे जाएंगे अभी आप कौन से सेमेस्टर में 8th सेमेस्टर में कोई बात नहीं अभी आपने इतना किया अच्छी बात है आगे आप इसको और इम्प्रूव करें और एम करेंगे तो और अच्छा इम्प्रूवमेंट होगा ठीक है तो आज आपको ऐसा लग रहा है क्या आपको यही क्वेश्चन है व्हाट मे बी द इम्प्रूवमेंट required for your presentation. It is very cloudy. कुछ भी नहीं समझ में तो कम लिखे ग्राफिक्स को ज्यादा यूज करें हम एक्सप्लेन ज्यादा करें जितना लिटरेचर में इतना टेक्स्ट कम होगा और जितने फिगर्स ज्यादा होंगे उतना अच्छा प्रेजेंटेशन ठीक है बेटर तृप्ति तृप्ति सोलंकी
Good afternoon to all. Myself, Ritu Kalindi from Computer Isaac College. I am here to present an e-poster on the natural anti-inflammatory agent. So, it is a biological response of vascular tissue to harmful stimuli such as pathogen, damaged cells, or irritants. Even in case where inflammation is caused by infection, the two are not synonyms. Infection is caused by the exogenous pathogen, while the response of organism to the pathogen is inflammation. There are natural agents which can be used against inflammation such as sandalwood, ginger, garlic, lavender, pepper. Prolonged inflammation persisting for weeks or months is known as chronic inflammation. Neutrophil accumulation is the hallmark of the acute inflammation, where the characterized by the presence of lymphocytes, monocytes, and macrophages are the chronic inflammation. Another hallmark of chronic inflammation is simultaneously process of tissue destruction and healing resulting in the formation of scar tissue. Inflammation may result from set of causes, endogenous and exogenous factors. Endogenous factors, the immune pathological response such as allergic inflammation and autoimmune inflammatory disorder. Endogenous products of tissue metabolism such as gout. Exogenous factors caused by the mechanical injury, physical injury, chemical injury, biological injury, and ischemic injury. Result of this study is that the use of medicinal plants instead of drug as active pharmaceutical agents for anti-inflammatory is more better and least side effects. The drug may person addict, but the natural agent do. So here I conclude that natural anti-inflammatory agents are alternative treatment for pain relief, especially for the long-term use. NSAIDs block the specific enzyme called COX, cyclooxygenase, used by the body to make prostaglandin. By reducing the production of prostaglandin, NSAIDs helps to relieve in the inflammation by the discomfort of fever and associated pain. Plants have capability to synthesize various forms of phytochemicals compounds as secondary metabolites, many of which have been identified and continue to be relevant in the treatment of inflammation. Thank you.
Okay, thank you, Tripti. So you mean to say that natural anti-inflammatory agents are better than NSAIDs?
Yes,
Why? How?
because they have least side effects and
NSAIDs also don't have so many side effects.
but the pharmaceutical agents are sometimes we can't prefer to the pregnant lady.
Paracetamol is safe. Many other NSAIDs are safe.
But some of which we can't be prescribed.
Uh, what in case of critical condition, critical pain, which type of analgesic do you prefer? बहुत ज्यादा अगर दर्द हो रहा है ना, तो उस केस में आप एलोपैथी पे जाओगे, आयुर्वेद पे जाओगे, क्या करोगे? नेचुरल एंटी इंफ्लेमेटरी एजेंट्स है। चलो इन्फ्लेमेशन की हम बात करें, तो किसी भी टाइप का इन्फ्लेमेशन जो क्रिटिकल स्टेज पे है, तो उसको ट्रीट करने के लिए हम एलोपैथिक ट्रीटमेंट लेंगे या नेचुरल एंटी इंफ्लेमेटरी एजेंट्स?
Allopathy
और ये कब काम आते हैं नेचुरल एंटी इंफ्लेमेटरी?
for the long term use.
Long term use and for prevention also. Sometimes we can prevent the upcoming possible disease. Okay. Name any examples of natural anti-inflammatory agents.
Ginger, garlic, peppermint, sandalwood.
Okay. But they are mild. Okay. अगर हम इनका एक्सट्रैक्ट निकाल के किसी फॉर्मुलेशन के रूप में देना चाहें, तो विच टाइप ऑफ फॉर्मुलेशन वुड यू प्रेफर?
Oiling, creaming.
Oh my God. You are talking about topical anti-inflammatory agent. Oil and cream. आपको क्या पर्पस क्या है यहाँ? टॉपिकली देना है उसको या आपको सिस्टेमिक एडमिनिस्ट्रेशन के लिए?
Tropical.
Topical. So you can give in form of oil and cream and gel.
Yeah.
Okay. And what type of diseases in which they can be used? Condition, inflammatory condition. Any examples?
In the mechanical injury or physical injury.
Only you mean to say wounds, wound healing. Teacher. Okay, thank you. Okay. Next presenter is Anjali Jain. Anjali. Very good afternoon to all of you present over here. I am Anjali Jain from School of Pharmacy, DAVD. I am here to present my topic, and I am currently doing M Pharmacy. I am in final year. So my topic is design, synthesis, and evaluation. Still, right now I did designing and of the antimicrobial and the antioxidant agents. So recently I'm focusing on antimicrobial agents. So we have taken both gram positive and gram negative bacteria and also the antifungal agents. So in gram positive, I did my docking in NVD. So first of all, the introduction, why I choose this antimicrobial agent, because we know that recently the very much use of antibiotic has been seen. So antimicrobial resistance has been seen, and class 4 antibiotics has also been resistant. So the need of new antimicrobial agent is required. And next, if we talk about the antioxidant agents, so we know that the major disease, which is cancer, and free radicals are mainly responsible for the ailment and tumors also.
So if we talk about the antioxidants, so that is why because they are the free radical scavengers. So that is why I think we major combination. And I have taken the cell ring, the major ring, which the functional molecule which is present over here is thiazole shiftase. So why I choose this is because thiazole shiftase are very much compatible with all the they give multiple pharmacological effects like antimicrobial, anti-tumor, antioxidant, and all these classes. And so first of all, next we will discuss about the methodology which I have talked for. So I did my doctoral studies on molecular virtual Docker. And apart from MPD, I did the 2D, 3D QSR study. So all of you might be knowing about the QSAR, which is quite, I think each and everyone might be knowing it. So quantitative structure activity relationship. So in 2D and 3D, first of all, I did the structure making on CAM, 2D and 3D. Then I did the energy minimization. Using this state software for my 2D, 3D QSAR. In this, I just took the IC50 value of the reported compounds. And in the software, what I did, leave one out method was used in this uh, out of total 90 compounds which are designed. So leave one out method was there in which 30 in the ratio of 70 to 30 I divided that. Just to know like which functional group is better for the human result, I'm going to synthesize further. So in this method, what we do, we take all the descriptors, topological descriptors, qualitative descriptors, and this tells about that whether lipophilic group should be there or not, or hydrophilic group should be there. It also tells that like electron donating or electron withdrawing group, which group is favorable for nitrogen studies and which will give the better outcome. So it gives a relation. And also I did two requests using Han's analysis and database. So in Han's analysis, there was a 0 1 table in the software with the test and training set. And the descriptors value should be like the pi value, which is the Han's constant, should be positive. It was coming, and the regression coefficient value is coming to 0.9, which shows that it shows a good relationship between the independent and dependent variables. Independent variable was my IC which I took in the reported papers, and the dependent variable was the activity which I need to see the topological activities and various factors which are affecting the compounds, the subsequent which I want to take to the logic study. Then further, our describing table over here, over there, the scores have been reported, and these are the various substituents at four positions of my shift base, and having a higher and the main group, which is the shift base at the center. And the topmost dog scores are taken because it shows the what exactly docking is. Docking is the interaction, the most favorable interaction between a ligand and the target. So the most highest dog score shows that it is the most stable and the energy is minimized. And this compound will be showing much more better activity. So in my docking results, I did the separate docking by taking the standard one such as cytotoxicity for anti-microbial positive aminopenicillin gram negative bacteria and for antifungal fluconazole. And these were I took three IDs over here. OHCT this is for gram positive, OB60, and OHCT the target is topoisomerase, OB60 gram negative bacteria target was DNA. And for antifungal, the target was 49 for so all these documents were compared to the standard drugs, and Classes was taken for gram positive and gram negative. I mentioned aminoparacetamide. So the dog score of these drugs were taken as standard, and the dog scores of 9 compounds were matched with each other. And three compounds showed much better activities as compared to the standard drugs. In gram positive, 18 compounds showed better activity, and in gram negative compounds, in antifungal activity compounds, we show the good highest output. So for that, we proceed to the synthesis of these top compounds, which shows a better quality of score. And I will go for the essays. And this is the procedure which I followed till now. Because 2D, 3D QSR helps us to take the functional which are necessary for the documentation.
And in studies that study hydrogen bond interaction and all those living interaction, would the validation also asked here is validation has been mentioned for the signal processing, in which it is there to validate the PDB and also our software which we are using. So, I took the PDBs and I brought the same structure in Android to me. And then I download all the structures and I match the interaction in the log score. This is what is called docking validation. So as to validate the software and the PDB which you have been using from protein data bank, access protein data bank. And so this till now I have designed the compounds which I will proceed for synthesis. Thank you.
Thank you, Anjali. Firstly, it seems good, but I should first guide you regarding the names you have written in the title. That your guide is written on the first page. It is your place actually because you are the presenting author and she has guided you. So you should write her name at last. Okay, presenting author always comes first. And he or she is actually the person who has, and at the last place it is corresponding author or your guide. Okay, so Tamanna ma'am's name should be at the last. This is the pattern you have to follow. And next, I'm a little bit confused about the methodology you have adopted. That firstly you have designed the compounds and then you have performed QSAR studies. So how you can perform the QSAR with the design compound without their IC50 values or biological activity?
What were reported literature, review of literature. So
It means you have not designed that compound.
no,
It is simply taken from literature.
no, these 2D3D QSA has been done by taking the PIC50 values of the previous mentioned paper. Then I designed after analyzing making QSAR SAMs
But for QSAR, if you are using, how many compounds you have used for QSAR? It should be more than 20, at least 20 compounds you need to perform QSAR. So firstly, you should mention this, okay? And then for docking study, okay, you have taken some compounds and you have performed QSAR. And then you have performed docking for all these.
On that basis, I designed my compounds.
Your own compounds on the basis of QSAR.
Yes, QSAR was showing that sigma has to be positive, bulkier group should not be attached. Okay. So on that basis, I designed my compounds.
Okay.
Electron donating groups.
How many compounds you have designed?
20 compounds.
Okay, fine. Then will all 20 were subjected to molecular docking?
Yes.
Okay. And how many compounds are you planning to synthesize now?
Top 10 compounds.
10. You will synthesize in how much time do you have to perform your future work?
4 months I'm having an
Oh, no, it's too short.
I started the assay techniques I'm doing, I perform EBPH for antioxidant and ABTS. So I'm taking for the standard with the ascorbic acid and kojic acid, these three standards for antioxidant. So I already performed these for the standards.
The standards.
On
And compounds are yet to be synthesized. What are shifts based?
Shifts base are imine moiety, which are the alkene group attached, like amide, we can say, carbon and nitrogen.
Okay, thank you.
Thank you. Basically,
Next is Priya Modi. Praveen Priya. Both right. So the e-poster presentation session is over from now. Over to lecture.
thank you so much, ma'am, for handling e-poster presentation and evaluation. So I would like to invite Mr. Sanket Khanna on the stage to present commendatory to Dr. Judy Pradhan. Thank you so much, sir and ma'am. Now it's time for our last scientific session. It is by Zua Shah, Medical Claim Officer, FMC Healthcare Limited, UAE. She received prestigious Pratibha Award by CM Kiran Kumar Reddy and Collector in 2013. She is silver medal winner for AFMI's 24th International Convention on Education and Gala Award in 2015. Eminent Scientist Award ICIB MDS in 2018. I would invite her on the stage for the presentation. Ladies and gentlemen, a very good evening to one and all. This is Bhusha with you. It is hard to put these feelings into words. Nevertheless, I am very glad to deliver my speech on such an important, momentous, and inexplicable occasion in my life. It is a huge honor for me to receive the award for Best Achiever. I am deeply honored by the Open Pharmacy Federation team, especially Dr. Mukram. He is a consummate professional with boundless energy, wonderful creativity, and extraordinary depth of cultivation and extremely wide experience. The subject of today's symposium, as you all know, is the era of pharmaceutical industries. I'm not a specialist in that area. You will be not hearing all that from me, which you have already heard from the esteemed professionals, the scientists, the researchers, and even the fellow students. Today I'm going to talk about medical coding. Have you guys heard of it? Anybody? I would like to add a comment from my professional experience. I am a medical claims processor for FMC Insurance Healthcare. It is one of the leading corporate companies in health insurance services with concept of cost-effective, comprehensive, and affordable healthcare for all. This is a humble beginning given by Dr. K.D. Hussain. He is an eminent personality renowned in career. Okay, let me tell you what's medical coding. So the medical coding, it is a process of translating important medical information into simple codes for the purpose of documenting medical records and for medical care. I'm going to tell you the story of medical coding. If you have uh, it was all started in USA. So there, a patient is normally called as member in insurance company. So a member will visit a hospital. Suppose a member, suppose me, I'm having fever. I'm suffering from fever. I'm having cold. I'm having cough. I'm dizzy. So I will visit a hospital and I will see a general physician or a GP doctor. I would see and I would say that Doctor, I'm having this is my illness, I'm suffering from this and that. So in USA, what basically happens is they are not paying any amount. When you go to the hospital in India, we are paying the doctor's fees and everything. But in US, you don't pay anything. You have an insurance card, you just keep the insurance card and that will be paid by the insurer, the insurance company. So in one of the cases, how they used to get the billing? How did they get the payment? The payment process was like the doctor will be there and there will be an underwriter. Underwriter will say this number is this, this age is this and that, and he will mention all the details and he will say that this number came and he took this services. Suppose, like in medical coding, there are codes. They are all security codes. So these codes they will put and they will charge the money from the insurance company. So here हुआ कि एक इंश्योरेंस कंपनी में एक डिपार्टमेंट रहता है उसको बोलते हैं कि मैं पेशेंट केयर डिपार्टमेंट तो पेशेंट केयर डिपार्टमेंट क्या होता है पेशेंट के पास जाकर पूछता है कि आपकी प्रॉब्लम क्या है उसको कैसे सॉल्व कर सकते हैं और उसको कैसे रिजॉल्व करेंगे तो एक बार एक पेशेंट केयर किया और जा एक मेंबर और पेशेंट के पास जाके बोल रहा है कि आपकी तबीयत कैसी है that lady said, I'm super fine. And then he asked, how's the doctor? They're saying that the doctor is the most amazing doctor in the world. You know, and so, क्या है ये doctor के पास? No, when I get, doctor didn't charge me anything. And he gave me bread, butter, and milk. ऐसा बहुत करता है तो उसने वो मेरी डॉक्टर को मुझे ब्रेड बटर मिल्क दिया then this patient is one of the show. What's this? It's something out of the box. Why the doctor is giving like this out of his own protocol? I'm not charging anything. So then उसने बोला कि can you show me your scar? And she's like, what scar? Then वो डॉक्टर ने claim के insurance company से that she has done a heart transplant. So that was completely fraudulent. So that leads to the birth of medical coding. तो उसके बाद उन्होंने ABC है that is a certified coder as per America. So you have to write the exam. And once you have written the exam, you are certified code. Then they have established the codes, the CPT codes. These codes are useful for the insurance company, for the providers. Providers is something like hospitals, clinics. Insurance to insurance company. So let me say more about medical coding. So as I told you, medical coding it is a process of translating important medical information into simple codes for the purpose of documenting medical records and for medical billing. Standard systems allow a more seamless transfer of medical records and more efficient analysis to track healthcare network of patients. So when this our PCA, there are so many medical codes, they must be तो ये मेडिकल कोडर कहाँ पर यूज आते हैं कहाँ पर आप लोगों ने सुना होगा कि ये मेडिकल कोडर है but we don't know what actually is medical coder. So these medical coders are useful in hospitals, in rehabilitation centers, medical professions, and insurance company and healthcare consulting services. So how can you become a medical coder? There are two exams. One is AHIMA and another one is AAPC. The most essential one is AAPC. AAPC is the American Academy of Professional Coders. So even if you write here in India or Bangladesh or anywhere in the world, you will be certified American coder. That's of great value. And one of choosing this topic to speak to you, being in India, there is no insurance, there is no medical insurance, no insurance company. But yet I want the students
to go ahead in a direction where they can lead a successful life. This one is completely a rewarding career, undoubtedly a career on demand. Training of this one, it requires occasional duration. You can even avail online classes or offline classes anywhere possible. It is based on flexibility and time, and that can be useful in the insurance company, and it is having a great pay. Believe me, if you crack this course, you will settle. If you work 8 years of your life as a coder, you are settled boss class. It will be done. That is of it is of that value. So students should go ahead and take this program according to me. And there for this there is a remuneration, a lot of remuneration, and this most profitable. So last but not the least, in a nutshell, I would say medical coding is a growing field. Plenty of opportunity are there. We are not aware, we are not aware of it. So for the students specifically, I would greatly advise them to go through this medical coding courses so that if you want to have a future in abroad, in USA and Gulf, anywhere you go, there is a medical insurance and there is a medical insurance company. You can avail it and have great. Thank you so much. Thank you for listening to me.
Thank you so much, ma'am, for your session. Now I would like to invite Mr. Vikram Chaudhary to present a memento to Zuha Shah. So now we are at the end of the conference. So we are going to start our valedictory function. In our today's session, we got a chance to meet and make network with many renowned personalities. We also learned a lot from their inspiring words, their knowledge, and their experiences. We had a very interactive panel discussion session, which increased our energy. Especially Dr. Anthony Cristo motivated us to make networks, which we usually avoid in conferences. So, and we also had a very three knowledgeable scientific sessions. Student also shows their knowledge in form of e-poster presentation. So overall, our today's conference is a huge success in terms of knowledge, experience, and network. Now, I would like to invite all our guests present here on the dais for the presenting award. I would like to invite Dr. Anthony Cristo, Dr. Shelly Chaudhary ma'am, Ankit Mehta sir, Vikram sir, Suhas ma'am, Nancy Sharma on the stage for the valedictorian function.
इधर है इधर है
So firstly, we will start with e-poster presentation award. For research category, first award goes to Anjali Jain. Can we have a round of applause for her? Second award for research category goes to Parul Dashora. Sorry, it's Parul Dashora. For review e-poster presentation, first award goes to Maya Pradhan. Second position goes to Satya Prabha. Congratulations to all the awardees. Now it's time to honor all our delegates present here. So first is Mayank Pradhan. Please come on stage and receive your certificate. Second, Pawan Vaishnav. Anjali Jain. Anjali Jain. Tripti Solanki. Muskan Haldia. Mohit Dwivedi. Amit Bajaj. Mr. Mahesh Menaria. Satyam Kandla. Pachi Gupta. You can come close to the stage. for your number. Mr. Banti Yadav, Priya Modi, Kriti Jain, Mr. Praveen Tawar, Adarsh Chebe, now we are going to honor our organizing committee, Mr. Alok Yadav, Mr. Ashutosh Chaudhary, Anjali Chauhan, so can we have a youth round of applause for everyone? Now, most amazing host, Ms. Ginji Arora. So, now I would like to invite our guests if they want to share any experience for today's session. If anyone wants to share anything, please be there for group photo. We are just going to finish in 5 to 10 minutes. Please stay. Good evening, everyone. So finally, the journey of good morning to good evening we have successfully completed. I would like to congratulate Vikram sir. Nicely, he have organized the conference, and I wish in future we again plan such type of symposium or conference in Indore. I would like to pay a very thankful thank from the corner of my heart to Anthony sir for providing me this opportunity to share the dice with the great personalities. Thank you so much. And congratulations to all the team. Thank you.
Thank you so much for the good. Now I request Sanket sir for your few words. Yeah, hello friends, good evening. I could see this energy which is converting into really productive actions. So in our time, we couldn't get these opportunities, but you people are taking advantage of this just because of Mr. Vikram. So I really appreciate this effort.
And thank you to invite me over here. And thank you, Tony.
So, as we started our for today's conference with so much of energy, so definitely we will like Mr. Anthony Mr. to end the session with their energy.
Let it complete his promise.
Yes,
Vikram,
he's going to
Vikram now leave.
after this. It's a surprise.
Yeah.
We all will get at them.
See, Vikram will only arrange. He's a master. Only thing is the state is not different up and down. Yeah. It was going perfectly fine. The third wave came. But only don't worry, we'll have a big one. Mostly, I think it might be Kathmandu or first two in India. That is what. Anyway, let others speak also.
I want to congratulate Mr. Vikram. His team. The team is really amazing and has done an awesome job. And Dr. Anthony for being here and super energy and transmitting the same energy and light into us and guiding us for the better future. Especially, I will talk about the host for her energy and the way she managed it. It's beyond my words. Thank you so much. Thank you for having me here. Thank you.
Thank you, everyone. कंटेंट में सब बोलती है की आगे कई लेकिन कंटेंट में देखो आपको डिफरेंट इंडस्ट्री प्रोफेशनल मिलते हैं डिफरेंट मेनली डिफरेंट कंपनी से आते हैं और डिफरेंट फील्ड से आते हैं आपको ये मिलता है तो आपको क्या करने मिलता है कुछ ऐसा नहीं कि खाने पीने के जाते ऐसा नहीं आप जो आपके साथ रहते हैं ये आपको आज पता नहीं चलता लेकिन फ्यूचर में आपको पता चलेगा कि जो मैं कंपनी चाहता हूँ कि कब से नेक्स्ट क्रिएटिव हुआ अंदर भी कुछ करने का ये सब जो लोग मेरे साथ है मेरा एनवायरमेंट में लोग घूम रहे मुझे भी एक फ्यूचर में इसे बनना है तो ये सब स्पिरिट आपको पता नहीं चलता लेकिन फ्यूचर में ये सब फील होगा तो ऐसे कंपेयर में सबको अभी सोचते हैं आपकी बहुत सा सोचेंगे खाना पीना सर्टिफिकेट ऐसा नहीं होता है इंसाइट की आपका ऐसा बिल्ड होता है पर्सनालिटी बिल्ड होता है और स्पिरिट आता है जो आपको फील करते हैं सब क्यों आपको सब देखे रहेगा और पर्सनली मेरा भी बहुत बात है कि इसको लेके एक्चुअली मैं भी से जब स्टार्टिंग ऑर्गेनाइजेशन स्टार्ट हुआ था मैं उनके साथ तो इसका भी स्टार्ट ही नहीं हुआ था मेरी स्टडी से हम लोग ने स्टार्ट किया उसको और जब हम मैं भी साइलेंट था काम नहीं कर रहा था मुझे आईटी सोशल मीडिया में थोड़ा सोशल मीडिया में मेरा तो उस टाइम में था उस टाइम में कुछ अलग सा नहीं था पता मैं एक बड़ा पेज था फेसबुक में तो इसमें कांटेक्ट की थी तो बाद में हम लोग ने ऐसा सोचा मैं सोचा नहीं था कि इतना बड़ा ऑर्गेनाइजेशन हो जाएगा लेकिन हम लोग ने स्टार्ट किया था तो बाद में धीरे धीरे जब मुझे बोला कि हम हैदराबाद में खास इंटरनेशनल कंपनी करेंगे सोचने से पूछा ठीक है मुझे खास मीटिंग था ऑनलाइन में था वो कॉल किया था तो कॉन्फ्रेंस में था तो कॉल बात की सब लोग तो ठीक है सोशल मीडिया मैंने आगे किया हम इतना सक्सेसफुली हुआ 700 प्लस टाइम खास इंटरनेशनल इसमें जो कंपनी को जानता था नहीं हम लोग ऐसा कैपेबल नहीं कि 700 प्लस लेकिन मुझे इतना मतलब मिला था सोचा नहीं था ऐसा हो सकता है ये ऐसा हुआ और हमारा ऑर्गेनाइजेशन मतलब और भी हमारे साथ ऑर्गेनाइजेशन को जा रहे थे हम लोग ने इससे आगे चल के लिए साथ और ऑर्गेनाइजेशन जुड़ गए आज कोई ये हमारा फोकस था इतना बड़ा फोकस था तो बाद में हम लोग ने कॉल
Now I would like to invite Mr. Ken Chaudhary to announce grand winner of today's function.
Five competitions over the 2 months. And around 500 students joined the competition from across the country. We are having the five competitions, including English competition, Stanford competition, Pharmaceutical competition, e-poster competition. Uh, Last one is the technical competition, story competition. So the grand winner among all of the competitions will be selected for the international intensive program that is offered by Government Pharmacy Services and Government Pharmaceutical Service Limited. For the night, we are providing the exposure to students for technical international experience over the 2 months of year. And the grand winner of international exchange program, he was not urgent here due to the exam. She was from Madurai College of Pharmacy, Madurai Medical University, Madurai. Miss Manisha Vedant. It was not present due to the exams, but from next 2 months, yeah, after the finishing of exams, he was going Japan for the internship. So, all the round of applause for the family. Now, I request everyone please be joined on the house for the group photo.
I will request after the photo session if you want to enjoy the bonus from please stay here. So now I would like to make the international statement what I think to all our youth collectors from Mumbai and future guidance. I would really appreciate them for sharing their knowledge and experience. I also extend my thanks to Mr. Vikram Chaudhary for organizing such a great event. It has cannot happen overnight. The field starts only weeks ago. It requires planning and the board also details. We have been fortunate enough to be a part of this team. And we are very motivated and dedicated towards the work. Our families are very motivated and their work is very job and result oriented. So, thank you so much, everyone. Thank you so much, our guests, delegates, participants. Thank you so much, everyone. Now, please now our bonus. These are basic, good things, knowledge,